Hello, class. This is the first of a handful of lectures about dopamine. First, we're going to understand dopamine as a neuromodulator, or more specifically, we're just going to understand what neuromodulator, what they are, what that means. There are a number of different neuromodulators in our nervous system. Dopamine is a very important one, and we'll be focusing on that for this series of lectures. But first, we need to understand what a neuromodulator is. So we talked a lot about neurotransmitters, but there are a few neurotransmitters that we consider neuromodulators as well. We've talked about GABA and glutamate, the primary inhibitory and excitatory neurotransmitters in the nervous system, respectively. And these are fairly straightforward neurotransmitters. They're released by the presynaptic neuron. They bind to receptors on the postsynaptic neuron, and they influence that postsynaptic neuron. They contribute to the postsynaptic neurons or postsynaptic cells membrane potential, right? And usually many signals are received by a neuron and they're integrated together. Right? So typically one glutamate or one GABA neuron sending a signal is not nearly enough to stimulate an action potential or meaningfully inhibit an action potential in that postsynaptic neuron. There requires typically integration of hundreds or thousands of signals simultaneously or in quick succession, one right behind the other. So neuromodulators, on the other hand, rather than directly contributing to the excitability or lack thereof of a postsynaptic cell, neuromodulators are chemicals that regulate a diverse population of neurons in the nervous system. And we'll be talking about dopamine, which has significant abilities to modulate nearly the whole brain. And so neuromodulators, they typically bind to metabotropic receptors to initiate a secondary messenger system, cascading signals that induces broad, long-lasting signals within the postsynaptic cells. And as we'll see, sometimes they can affect, as we go on in our videos, we can see dopamine can do more than just influencing one single neuron that it synapses onto. So let's talk about specific examples of neuromodulation, of which there are many. So as we go through this list here, I have on the slides here dopamine, the neuromodulator, right? But in fact, this neuromodulation that I'm talking about, we will be going over examples that aren't dopamine. And so it's important to know as we go through these cycles, even though this is in the context of building up to understand what dopamine is doing, specifically in our own reward pathway, that this portion right here really is more general than just dopamine, right? So this is neuromodulators in general, right? Of which dopamine certainly is an important one. So keep that in mind, that these examples here are not specific to just dopamine. So first, there are many different types of modulatory effects. This first broad one that I'd like to go over could be described as altered neuronal excitability. So neuromodulators have the ability to influence the excitability of neurons, their intrinsic excitability, influencing their propensity to generate action potentials, either making it easier or making it harder. And we're all very familiar with the mechanisms of action potentials and what that requires. It really centers around really two very important voltage-gated ion channels, sodium and potassium. And we've learned about these little machines having specific thresholds for activation when they reach a certain when they're exposed to a certain membrane potential. They open, and when they're exposed to a certain depolarizing potential, they can close or become inactivated. And we also learned about their ability to conduct their specific ions through the channels, Right? And though we didn't talk about this in any great detail, they usually, the properties of the channels themselves influence the rate of conductance. All right, the amount of current in the form of ions that come through the channels are driven by both the concentration gradient, the electrostatic gradient, of course, but also the intrinsic properties of the channels themselves. Right, a limiting factor of how many sodium channels that can come through, for example, at a given concentration gradient and electrostatic gradient. That limiting factor is the intrinsic physical nature, atomic nature of the forces in that channel itself, based on the structure of the channel. And so that's important to keep in mind for this first example here. And so we're going to be talking about acetylcholine and how acetylcholine can modulate the excitability of neurons in the central nervous system, affecting their responsiveness to incoming signals. So we'll see about how acetylcholine, more than just triggering an action potential or starting that action potential event happening in the postsynaptic cell, we're going to see how it can actually change the cell to make it more sensitive to future action potentials. So let's take a look here in this drawing I have here. Okay, so let's take a look here at this example of acetylcholine. What we have here is our presynaptic neuron on the left and our postsynaptic neuron on the right. And so because this is a postsynaptic neuron, we're not in the periphery. This is not a neuromuscular junction. Rather, this is in the brain. And we can see that acetylcholine has been released from its vesicle, right? So this presynaptic neuron has received an action potential, and it's released its neurotransmitter, right? So acetylcholine. And acetylcholine will bind to this receptor that's on the postsynaptic neuron. Now, what is this receptor? Well, you might be able to tell a little bit of information by looking at it. What we have here is a receptor with 7 transmembrane spanning domains. And this little blue bit here is our what's known as a G protein, which we've talked about before in class. So this is a G protein coupled receptor. And what kind of receptors do we call that in the context of neurotransmission? We call these types of receptors metabotropic. And what is our class of metabotropic receptors for acetylcholine? These are our muscarinic receptors. So this is a muscarinic receptor that we have here. And so what it does, instead of being unlike ionotropic receptors, in which the receptor itself is also an ion channel, this is going to start what we would term a signal cascade. So this protein will actually, G protein will actually disassociate. And it's made up of multiple protein units. And I'm not going to get into the details of all the steps in the signal cascade. For one, I don't know them all, and we're going to be going over a variety of different steps. So we'll just talk about them, the more salient parts within this signal cascade. So in this specific example, where I want to highlight in the ability for a neurotransmitter to modulate neuroexcitability, this signaling makes its way all the way near the nucleus, in fact, right? So we're sort of taking I'll try to keep my my arrows the same size. Despite this picture here, you can imagine this going all the way through the dendrite to the soma, where it's going to interact with the endoplasmic reticulum. Right, so this is a structure that we find on the near the nucleus. So right here, we'll say that this is our nucleus. And I'll use red for our endoplasmic reticulum. And so we have two general types. All right, this one here on the outside, this is our rough endoplasmic reticulum. This is a structure that's involved in a number of different things, primarily most well known for its synthesis, folding, packaging of proteins. And this is our rough endoplasmic reticulum. Let me write this out here. So we have our endoplasmic reticulum. And this is often referred to as just the ER. Right, and this one is I'm just going to label this one rough. It's the rough ER. And we also have what's known as the smooth ER. So I'm going to just draw this without any of those little blue dots. And this can be associated with the rough ER, or it can actually be free in the cytoplasm inside the cell. And the smooth ER has proteins in them, and these proteins bind what I'm going to represent as the little green dots here, bind calcium ions. So these green dots here are calcium ions, chock full in our smooth endoplasmic reticulum. And so our signaling cascade makes its way to the smooth ER, and what it does is it signals the release of our calcium ions. Now, our calcium ions, in addition to being a charged particle that can be used to influence the membrane potential of a neuron, it also acts as a signal messenger, a secondary signal messenger. And so this can go on and facilitate other metabolic processes within the cell. Right? And these processes often involve a lot of phosphorylation, right? So transferring energy from ATP to other proteins to change their structure, to influence what they're going to do, what they're going to affect. And so this signaling continues. I just can't get this down. Here we go. Continues signaling. All right. I'm sort of running out a little bit of room here. So let me expand, give us a little bit more room here. Right, so the signaling continues. And what this specific signaling cascade is going to give us is some interesting changes. I'm going to talk about these changes in generalities here and not get into the specific mechanisms, but it's going to affect two different types of channels, which I'll color code here. Let's make it, we'll make them red and blue. All right. And so our calcium, what started as our acetylcholine binding, resulting in the increase, the liberation of calcium from the smooth endoplasmic reticulum, and that calcium going on to influence signaling through other proteins, eventually makes its way back to these two channels here. And so we have sodium, and these are voltage gated. Voltage gated channels. And we also have our potassium voltage-gated channels. So I'm just going to say VG for voltage-gated, VGC, voltage-gated channel. And so what this does is it modulates these voltage-gated channels. And so we know that our channels allow the input or the flow of sodium into the cell, right? And then also the flow of potassium out of the cell. And because of the intrinsic properties, the intrinsic structure of the channels, there's sort of a limit to how many of those ions can flow through. Of course, it's driven by the concentration gradient, the electrostatic gradient that that you're very familiar with. That can influence how fast it flows through. But this channel is only so big, so to speak. It can't let an infinite number of sodium ions through or potassium ions through. Right? There's an inherent limit, and that inherent limit is based on the structure, the protein structure of the channels themselves. Well, one thing that this signaling cascade can lead to is actually an increase in the conduction of these sodium channels. So it changes the structure of the protein channel in a way that allows more ions through, so it becomes more sensitive. So I'm going to put up here, we're going to talk about the two things that it does. So that's one, it changes the conduction. All right, the conductance. Of ions. And two, it can change the gating properties. And so in addition to altering how many ions can go through, it can also alter when they turn on. So we know normal threshold for opening these channels, right, is about negative 50 millivolts in the examples that we went over in class. So whenever the membrane potential depolarizes to negative 50, this triggers the sodium voltage-gated channels to open. Also potassium as well, but as we learn, potassium is much slower, so it gets a delay before things really start moving. But with sodium, it opens and floods in very, very quickly. And so what we can actually do here through signaling is we can actually change this threshold. And so this signaling may increase the threshold. And so this may mean that we have to now depolarize to negative 40 millivolts, just using as a relative example. I don't know if this is actually, if 40 is a real number in which it can change to. But the point is that you can make it harder for them to open, or you can decrease the threshold. And say, like, instead of negative 50, it's negative 60, which, if you remember in our examples, typically we talked about the membrane potential being the resting membrane potential equaling negative 65. So in this example where we've lowered the threshold, we've made these channels more sensitive now. So it doesn't take nearly as much depolarization to trigger them. And so in this example right here, where we've lowered the threshold, we've created this neuron now has, so to speak, a hair trigger on being able to produce an action potential. And not only is it more sensitive now when it does open, it can allow more ions to flow through than normal. And this is just due to all the way back here with the initial signaling of acetylcholine, multiple steps in between releasing calcium, which leads to other signaling, which will alter these channels. All right, so we can actually change our, right, and remember what we started talking about here, this specific type of neuromodulation, the neural excitability, which is a really cool example. I mean, it was already neat to figure out some of the mechanisms here. We're like, oh, wow, neurotransmitters can actually open ion channels and dictate whether or not an action potential is more likely to fire, or can perhaps hyperpolarize and influence, inhibit the neuron. But it's much more flexible than that. These neurotransmitters, if binding to the right type of neurotransmitter, namely metabotropic transmitters, they can do so many more things. They can increase the excitability or decrease the excitability of the neurons. So they can not only neurons turn on or off the neuron, it can actually change some fundamental properties of the neuron itself. Okay, let's take a look at another example, different type of neuromodulation and another example. Okay, so now I'm going to talk about a different general class of neuromodulation. We talked about neural excitability, how a neurotransmitter can influence how sensitive or insensitive a neuron is to propagating an action potential, starting and propagating an action potential. And so that was all about signaling, changing how a neuron sends signals within the neuron itself. And so now we're going to talk about a class with many different examples of a synaptic efficacy. So neuromodulation that can alter synaptic efficacy. And so they can modulate the efficacy of synaptic transmission, which influences the strength of communication between neurons. So in the previous case, a neurotransmitters affected how action potentials were propagated, making them more sensitive or less sensitive. In this example, we're going to see that a neurotransmitter has the ability to change the way a neuron responds to information from other neurons. Right? And so we can see through this modulation, we can change the efficacy, so the ability for the neuron to produce a signal from a neurotransmitter. And so the efficacy of synaptic transmission, right? How good is the presynaptic signal at triggering the postsynaptic effect? And so let's take a look at our first example of this type of neuromodulation. We're going to talk about facilitation. This enhances or facilitates the transmission of signals between neurons, making the postsynaptic neurons more responsive to excitatory inputs. And in this example, we're going to take a look at serotonin and how it can facilitate synaptic transmission, making it easier for excitatory signals to induce a postsynaptic response by liberating stored calcium, modulating, which in turn will modulate ion channels. So let's take a look. All right, in this example, we're going to take a look at how serotonin can induce facilitation, a specific type of neuromodulation. So in our presynaptic neuron here, we have vesicular excitosis, and this, in our example, of course, is serotonin. Serotonin is also known by its chemical name, 5-hydroxytryptamine. Let's see, I think my spelling's a little off here. Hydroxy. There we go. Hydroxytryptamine. And often you just see it shortened to this. 5-HT, synonymous with serotonin. Okay, and so serotonin has a number of different receptors that it binds to. In this example here, for this facilitation explanation, it's binding to what we call simply 5-HT2. And indeed, this is like the other example, we're going to be looking at nothing but metabotropic receptors. So this is a metabotropic receptor, right? So we know that a signaling cascade is going to be involved. And for this one specifically, introduce a specific explanation here of one of the first steps. All right, so we've got, I'm just going to draw this little membrane bound protein here. And this is a phospholipase C. And what does phospholipase C do? There's the C. This is a membrane-associated enzyme that cleaves phospholipids, right? And so these are molecules that our membrane is composed of. And the output of this enzymatic reaction, the components of those phospholipids can act as secondary messengers. And so what we have here is through serotonin binding to our 5-H2 metabotropic or G-coupled protein receptor, we have a signaling that signals the phospholipase to induce its enzymatic activity, which starts a signal cascade again of secondary messengers. And in this example, the specific example, it liberates calcium, once again, stored calcium within the cell, which induces further signaling. And this is going to make its way to channels that are important in our postsynaptic neuron. Two of them in this example here. So I'll stick with green for one of them. The colors aren't going to sort of carry over from drawing to drawing, so the colors will start from scratch again. I think green and the last one was sodium, but this is actually calcium now. Calcium, and these are voltage-gated calcium channels. And then also another receptor over here, and this is our NMDA receptor. And do you remember what NMDA responds to? Right, it responds to a different neurotransmitter. So I'll draw here in blue, glutamate. Right, our primary excitatory. Right, and so this neuron, right, most neurons typically just release sort of one flavor of these primary neurotransmitters. You typically don't have a neuron that releases glutamate and GABA, which is our main inhibitory neurotransmitter, releases one or the other. But one neuron can receive inputs from a whole variety of different neurotransmitters, right? So this neuron can receive serotonin and glutamate, right? If it has synapses from, right, which requires synapsing with multiple different neurons. And so this signaling cascade here will, in this example of facilitation, and let me just write that up here so we don't lose track of it. It can, to use a fancy word, potentiate. And what that simply means, you can think of it as just, it makes it stronger. And so what it does is similar to what we saw before in neuroexcitability and how it affected the voltage-gated ion channels associated with generating action potentials. It can, in the case of our calcium channel, it can influence perhaps the conductance of calcium into the cell, or it may change its sensitivity to opening and responding to membrane potentials. Right, and the difference here is we're talking about synaptic transmission. And so by changing these calcium channels, these are involved in responding to the signal of a neurotransmitter. So if you can enhance or potentiate calcium voltage-gated channels and make them stronger, then the binding of a neurotransmitter such as glutamate is going to give us a stronger postsynaptic effect. And so whereas, to make it very simple, whereas before, let's say we needed two glutamate signals, so two glutamate receptors to respond to a signal in order to give us a postsynaptic action potential, whereas we needed two of these before to bind. If we could, and what they do, right, is they allow sodium to come in, which in turn changes the membrane potential enough to open up a whole bunch of calcium channels. And so, but if we make the calcium channels, say, more sensitive, then we might not need two. We might just need this one. So you can see how this will make it more sensitive. It facilitates this neurotransmitter communication that glutamate would do. In addition to making calcium voltage-gated channels more strong, it can also increase or potentiate the glutamate channels as well. And so it can make it more, it can make the NMDA receptors more efficient or efficacious for glutamate. Right? So it can make it more efficient. I'm probably botching the spelling here. Yeah, indeed. Oops, doing it again. More efficient or efficacious. And I'll probably botch this again, but I think you know what I mean, efficacious. And so by making the glutamate stronger, it can increase that signal as well. So the postsynaptic neuron, by strengthening calcium and NMDA receptors, in this case via serotonin, it makes that postsynaptic neuron more sensitive to presynaptic signals, right? It's more likely to sort of turn on, so to speak, with a smaller signal if you can facilitate it. Okay, let's go on to our next example.